Welcome to this session on the sample management lifecycle in action, stages, stakeholders, identifiers, and opportunities, not to be confused with lifecycle in action. To start the session, I would like to acknowledge and celebrate the first Australians on whose traditional lands we meet today, and pay my respects to the elders past and present. And our agenda for this session is that I welcome you and introduce you to the session. And then we have four presentations looking at different perspectives of the sample life cycle, how researchers interact with it, how collections curators and the repository managers interact with it, what the data and information specialists see, and the role of infrastructure providers and publishers. And then this will lead into a discussion where we will discuss what the state is of what we have today and where we see gaps and potential for better alignment with our processes, and then wrap up this session. So from my perspective, samples are the anchor of the scientific knowledge graph in reality. There's a lot of abstract things we can build and connect, and but it's really the ground truth that anchors a lot of our argument in the physical reality. So effective sample management is really essential to ensure the integrity, reproducibility, and openness of research across diverse disciplines. And it's complex. It can be at really large scales. It can be really diverse. The sample collections are growing, and we need robust interoperable management strategies to make, and this is becoming increasingly urgent. Persistent identifiers play a central role in addressing this challenge, and they need to connect to samples, they need to connect to the related data sets, the publications, instruments, and the contributors. And to implement this requires coordination between the multiple stakeholders that are involved in the sample lifecycle. So all of these stakeholders, researchers, collection curators, data informatics specialists, infrastructure providers, publishers, they all contribute in different stages of this sample life cycle. But what we all know is that there are gaps. There are gaps in standardization, it's not always interoperable, and a lack of best practices sometimes also is a blocker in effective sample management, linking our ability to make all those cross connections that we ideally would want to see between disciplines, in the disciplines and in the repositories and the rest of the scholarly knowledge graph. So as our first speaker, we have Natalie to talk to us about sample stewardship for researchers, realities, needs, and opportunities.
Perfect. Thanks so much, Jens, for the intro, and thanks to you all for attending the session. My name is Natalie Raia. I'm a postdoc in the College of Information Science at the University of Arizona in the United States. I also serve as the User Experience and Community Engagement Researcher for CSAR, which is a IGSN allocating agent and the birthplace of the IGSN. Today I've been asked to speak to the researcher view on sample stewardship. This is personal to me. I'm a geochemist by training, and sample stewardship and sample data management was actually my inroads into the data space. So it's very nice to be here talking about that topic. But I also wanted to emphasize that the perspectives that I'll be sharing really are underpinned and deeply informed by a number of other recent community engagement efforts the Sampling Nature Research Coordination Network, the Earth Sciences Information Partners Physical Sample Curation Cluster Working Group, as well as the iSamples project. These are all US-based initiatives, but it's very nice to see work on the international stage here on display this week towards these challenges as well. So I just wanted to touch on the researcher realities. So we know that sample metadata are diverse. They are core to many different scientific disciplines: archaeology, geology, hydrology. I'll be showing my geosciences bias here, but insert your favorite picture of samples here: water, soil, fossils. These metadata are often collected in unstructured and analog formats, like the field notebook shown here. And sometimes they're being collected under a time crunch. If you're out in the field, you gotta keep moving to get your work done. The same could go for synthetic samples as well, experimental samples. And sample identifier practices vary widely. They're highly heterogeneous. They vary from researcher to researcher. They may be passed down through kind of mentorship models, and they may not be aligned with best practices that exist in fields such as library and information science and archival studies. And then to add to this, researchers may not be thinking about data reuse or sample reuse at key moments of sample metadata collection. And then finally, samples, subsamples, and related research products can proliferate very rapidly. So you have these hierarchical relationships between samples, derived subsamples that have been processed in some way, and then all of the data sets and other research products, publications that derive from that. And so linking these becomes something that can be quite overwhelming and complex for a researcher who has just kind of very practical needs to track these things. More bad news, perhaps. There's little to no institutional or other incentives for researchers to think mid to long term about physical sample management. And many researchers, particularly working in the long tail of research, may not have access to a long-term physical archive for the samples they collect. Concepts of sample ownership may also hinder motivations to share samples and sample metadata openly. And I want to underpin that ownership does not equal stewardship for the vast majority of researchers. So they may feel these are my samples, I need to keep a hold of them to do work on them, but then there's a lack often of that management and that curation of both the physical material and the data. Researchers may be unsure of which material to preserve, and they may have little to no awareness of sample persistent identifiers and the why of using standardized metadata to enable discovery and interoperability. And so from the researcher needs side, and again, this is underpinned by these ongoing community efforts, we hear needs expressed for institutional and funder requirements. We've actually had researchers say, I wish we would have the stick, please give us the stick, or other forms of support, so carrots such as institutions fiscally putting some support into that curation work and storage. We need clear examples of the benefits of sharing and reusing samples. We also need tools to support streamlined, standardized collection and archival of sample metadata, and we'll hear about some of those ongoing efforts today. And researchers have also expressed needs for guidance in best practices for sample and sample data stewardship throughout the research lifecycle. And so that's what I'm just going to touch on briefly in the remaining slides. This is a new paper out earlier this summer that was work conducted over almost 5 years with the Earth Science Information Partners, specifically the Physical Sample Curation Working Group. You can see the title Earth Science Information Partners, but these are interdisciplinary authors coming to the table, interdisciplinary efforts, so they're broadly extensible, we believe, to other disciplines. The underpinning work, I'll show the recommendations in a moment, but the underpinning work here looked carefully at 4 core use cases for researcher needs to tracking sample use. And they focus on the needs to publish and cite a large number of samples and associated data and metadata efficiently. The need to provide credit for those involved in physical sample collection curation, particularly in order to demonstrate the value of investing in collections and putting people investment towards doing that work. The need to track the use of sample data that's been generated by laboratories. And then the need to connect related interdisciplinary sample data, metadata, and other research outputs. And so that's really the core of where this PID element comes in. The recommendations are shown on the right, and that's attached and cited in the paper that I showed on the last slide. There's also more of a visually pleasing companion quick guide that's there as well. But we break this down into 4 key steps. These guidelines are specifically aimed at researchers, at authors. Here's the step-by-step guide from the moment of sample collection or generation all the way through to publication of your data and publication of your papers. Here's what you do at each step. Here's where PIDs play a role. Here's the why and the how. And these are designed to be iteratively versioned as infrastructure and stakeholder practices continue to develop, which they are developing rapidly, I would say, right now. So we're currently on version 3. And then just to kind of wrap on some opportunities, this is a summary figure. I call it the rat's nest figure that is in the paper that I linked there, and I have it here as well in the QR code. We examined the stakeholders in this space. I think it's a highly relevant slide for this session structure. And then we broke down the socio-technical challenges that exist across the ecosystem for kind of enabling and leveraging sample PIDs to the best ability. So in orange triangles, we see social, human-enacted practices and workflows that need to develop. On the red side, which you'll notice is more on the right side, these are technical infrastructure needs. So everyone here has a part to play. I'm sure everyone in the room can find themselves somewhere on this graph. And so in the paper, we present actionable next steps for each of these stakeholders. And we don't have funders on this particular graph, but we've had conversations about them as a stakeholder as well. And so, yeah, just to wrap, I've kind of touched on these two, and then the remaining presentations, I think, are going to speak to other components of this graph. Thanks.
Thanks, Natalie. I think we'll move on to the next presentation and keep all the questions for the discussion at the end, so we get the different perspectives now and then reflect on them later. So I'd like to invite our next speaker, Kerstin, Lehnert.
All right. Whoops, I didn't know you are so big and tall. So I'm Kirsten Lehnert from Lamont Doherty. I'm the director of CSAR that Natalie already mentioned, the System for Earth and Extraterrestrial Sample Registration, and which I started 22 years ago about now. So it's a long-term effort that has led to the IGSN and other stuff. And I'm going to focus in my presentation on the institutions and the people who actually manage the physical samples as their job, the curators of sample repositories and museums. And the reason I feel confident in doing that is that CSAR has actually, over those 20 plus years, interacted a lot with curators of sample repositories and museums. They are the biggest users of CSAR. As you can see on the left side are just the total number of samples from those facilities that are in the CSAR catalog right now. On the right side, you can see how many samples are being registered these are just the 5 top facilities that use it. And I would already like to point out on the right, you see that it's primarily the sample repositories that registers only one museum, and that is because museums have fairly stable collections and only receive smaller numbers of samples, while the sample repositories have ongoing sample management and accession of samples. Just to emphasize that again, I wanted to distinguish these two stakeholders. Museums, the main task for them is to preserve and then get the samples displayed in exhibits and so on to the general public, while also making them to some degree available for research. They're really there to safeguard heritage, be it on the cultural side, be it on natural history, and so on. They operate with formal curation policies and procedures, but on very different scales. The big museums that I showed was the Smithsonian Institution of Washington, the Natural History Museum, that has absolutely fantastic, huge collections, but there are also small museums on local scales, even privately funded museums. In contrast, or it's slightly different at least, the sample repositories are there to store and manage and disseminate samples that are needed for active research. And so they are usually operated, there's also a large variety, but they usually operated as research infrastructure that is often funded by research, science foundations, and other research funders. And operated as part of universities or other types of research institutions. And something I wanted to add there is they often funded by time-limited and competitive grants, so they struggle with resources and with the persistence of their operation. And just 3 examples of repositories that I've come to know, and that already shows the kind of difference of their scale, and I'm coming back to that. And so, but there's an overarching purpose for museums and sample repositories. Both ensure long-term preservation of the physical objects in their collections, and they have to focus on the actual physical aspects. They need to manage space, they need to manage the storage of the samples, make sure that they don't degrade or anything, that they stay in controlled environments. They need to keep an inventory, obviously, of everything that they have. But then, again, the sample repositories at a bigger scale, but museums as well, want to facilitate access and use of the samples, because that is their mandate. And they are very keen on understanding or tracking the use of their samples, the impact, the societal impact of educational activities, and a lot of what their displays and their outreach activities are doing. So in order to achieve that, they need to disseminate information about the collection. They also work on preparing subsamples for research to be, the subsamples being sent out for requests from researchers. But on the other hand, they also acquiring and accessioning constantly new collections and new samples. Again, there is a lot of diversity, and I just copied here. This is a repository at the University of Nebraska in the US that actually says they're also collecting samples. That is not the case for many of the sample repositories. They just take in samples that come from field work of researchers and so on. There were large differences in organizational structure and policies, and specifically emphasizing there's a lot of variety in the resources that are accessible for the curators. And that really determines, I think, their role in this world of the digital sample management, because very often there is no support for IT, for databases. There's no expertise that they can rely on, and they themselves are overworked. Much of volunteer effort is still going on in museums, so that limits, I think, what we can ask and expect in the digital management from curators. These are the examples that I showed previously, and they just give you a feeling. I mean, the biobank in Switzerland is obviously well funded by the government and very well established, while the US Polar Rock Repository is one of these facilities that limps along with 3 to 5 year grants, has built its own database, then the developer left, and there's a lot of struggle. And I also know how they desperately reaching out to investigators working in Antarctica to send their samples to the facilities. So that is another big difference between something like the biobank, where there are mandates to send the samples to the facility, as compared to the repositories that really need to get investigators to use them. This was just funny when I yesterday went online to get a picture of a curator in action. The first thing that came up was this Lego doll showing sort of the stereotype of a curator, and then seeing, who are the curators today. This is the new curator of the Gems and Minerals collection at the Smithsonian Institution. There's some, I think, work that we need to do in re-emphasizing who these people are. But what I've learned in the interaction with curators over time is what they care about is to effectively promote their collections to users, to funders, to justify the money that is being put in. They really care about the physical safety and security of their collections. They need to be streamlined in cataloging the samples. It needs to be efficient, what they do. And especially this comes when it comes to legacy collections. When they get a collection of 1,000 samples sent by some researcher, they struggle to actually get that translated. And they want to align with existing community standards and practices to link up with other facilities and become part of global search engines and so on. So from that, I deduct the curator's role as digital sample stewards to be really a critical role. Museums and sample repositories need their samples to be fair. They need to publish the samples online so that they can be found and can be accessed. There need to be landing pages and so on. And increasingly, access to the metadata of samples in machine-readable format is critical. And in that respect, I see I think the curators understand increasingly that registration with persistent identifiers in systems that make the metadata more accessible is a huge benefit for them. Again, enabling community metadata standards so that the samples can be better discovered, but also integrated with other samples or reused with the right level of understanding, description of the samples, so that the samples really have a long-term impact. They also understand that they need to move from local naming protocols to persistent identifiers. The good thing is that what worked for me in explaining the benefit of using IGSN at the time was that you don't have to rename all your samples. That's not the problem, leave your name on the labels. It's about having a resolvable identifier that goes along with your own name, and that has really worked. And the reason why they understand the benefits of persistent identifiers is that the linkages of the samples to the data, to the publications, enables them to track the use of the samples in a more streamlined way, really. Last slide, I just wanted to emphasize some of the obstacles that the curators face. In general, curators are reluctant to make changes to their workflows, to the protocols and naming and so on. And that has to do with the resource limitations of staff effort or the lack of IT infrastructure and support that is needed to make changes. And the disruption of established procedures and operations also impacts, their interaction with users, that users need to be informed about new ways of doing things. Again, few curators are trained as digital sample stewards, and I think that's something we really need to work on to provide educational opportunities. And helping them convince the researchers that there are new ways to deal with samples and establish new practices. They really don't want to lose their users and push them away by saying, you need to register every sample and so on. Yeah, that's what I wanted to say. The next one.
Thank you, Kerstin. Our next talk is by Anusuriya Devarajum, who will look at the interface of what we've already heard about the researchers and the collection and the end-to-end sample management.
Hi everyone, my name is Anu. I'm a data architect at CSIRO Australia under Mineral Resources Discovery Program. Today I'm going to share with you I actually led a strategic project funded by the Discovery Program on end-to-end sample curation and management. I'm going to share with you the result of the project, most importantly, what are the lessons learned from the project. So before we started the project, there are two challenges when it comes to sample curation and management, and you can categorize them into digital access, and the other one is physical curations. Prior to this project, there was no central catalog of system available through which the researcher or scientist can easily discover samples beyond their project unless they know the project lead or they know the technician who have prepared the samples. So there's no way people can find samples from other projects. The second challenge is related to physical curation. So within our program, samples are handled on an ad hoc manner on a project basis. And these pose several issues. For example, as you can see in the pictures, there's no proper procedure exists on how the sample should be collected, how they should be recorded, and how they should be packaged and finally archived in a storage facility. So over the time, as you can see in the picture, the boxes in a pallet collapse over time. We often find sample in under someone's desk or in a cupboard, and there's no label. People don't know who owns the samples. So to actually tackle these two digital and physical challenges, we actually, the program funded the one-year strategy project. We have successfully delivered the project. It is now being adopted in the program. Now we are at the second phase of the project, which started August this year. So I'm going to share with you the three main components that led to the successful delivery of the project. So these are three main components. All of them are very important to deliver end-to-end effective sample curation and management that include technology, people, and processes. All these components we need to make the sample curation successful. I'll start with the processes. So we have developed guidelines. This is a practical guideline that contains recommendations and end-to-end workflow, how that samples should be managed, starting from project proposal stage, and then collecting the sample, preparing samples, sending them to the lab for analysis, and finally archiving them in a proper storage facility. Within this guideline also, we clarify what are the roles needed, what are the responsibility comes with the roles, what are the protocols to label the sample, how do you handle hazardous samples. So basically all the requirement and recommendation, we have developed that in this practical guideline. We also have developed several standard metadata templates. As you know, our samples are diverse from rock, vegetation, soil, and groundwater. So we have a standard template to ensure standardized, to streamline data entry, also a lot of control vocabulary as well. So the important part of that practical guideline is this end-to-end sample curation workflow. So basically, it covers all the stages of sample life cycles. And for each of the stages, we have actions. And it's quite small to see, but basically for each of section, we have the role and what are the actions should be done. So basically, this is like a Bible in the sense that anyone starting a project, the immunoexpression project that requires sample collection, they should be able to follow this, starting from project proposal collection up to archiving that in a facility. And these developed in collaboration with laboratory technicians. So it's not like a top-down. It went through several iterations, and it has been improved through this input from the scientists as well as laboratory technicians. All right, the second part is the technology. So we have developed a system called CMR Specify based on an open source tool called Specify. Specify is an open source primarily developed for managing biological collections. So our contribution is on adapting Specify to meet geoscience sample requirements. The system actually provides a lot of different functionalities, starting from how you can search the sample with a simple keyword search and more like a structured search using the query builder. You can also record information of the sample using online form, but if you have more than 100 of sample, you probably use the bulk upload function as well to catalog the samples. There's also function to generate report. Labels, there's also function to loan the sample. And then when you return it, how do you record it? So basically, it covers all the basic function we need for physical sample management. Just to give you some specific, just assume I'm a curator in a project. At the end of the project, I just go to the system. I find all the sample related to my system by a project name. And then I have my result display on the right side. As you can see in a tabular, I can also see the map. And then from there, I can also generate a report, which I can attach to the project report. This PDF also, you can print it out, you can also include it in the sample container. And I can also print label and attach to actual sample. So the system support all this. This is just a specific use case how the curator will be used the system at the end of a project. Right, this is the most important one. People. I think Kirsten already mentioned something about how important to have that digital sample steward. It's a new name I'm hearing. So basically, the successful sample curation and management require roles and clear responsibilities. So if you're starting a project, for example, the project lead should ensure appropriate resources and also materials to actually package the samples. And the curator, mostly the technicians, these are the people who prepare the sample. So they should know how to use the system, how to catalog the sample information, how to use the system to generate report and labels. As I say, the project team is one of the successful reason why this project was successful is that because the team is a very diverse team consists of engineers, scientists, and laboratory technicians. And we also, as you can see, we have a lot of engagement activity as well, like a user manuals. And we also have a help desk, so people who want to start the project, they don't know how to get on board, so we can provide that support as well. And I think most important, is the leadership endorsement. So the guidelines and the tools we have developed has been endorsed by the program leadership. So that really helped the wider adoptions. So key learning. So effective sample curation and management go beyond a technology piece. And you need to have that people component processes, which is basically the practices and technology all come together. The second one, you need to plan the sample curation and management early from the project planning phase. Otherwise, you will end up a backup of samples at the end of the project. We have a lot of legacy, because we started the project 1.5 years ago. When we started, we had a lot of requests to actually create legacy collections. Someone from 70, where I haven't born even. So it was like a lot of legacy collections. So there where we actually prioritize it. So the priority come in terms of ownership. Anything that's owned by CSIRO, we actually put effort to curate it. Anything is not owned by CSIRO, we send it back to the company or state government, for example. And we also prioritize it based on the level detail of curation depend on how important the sample will be used in the upcoming project. Some sample, if they really want to use in the next project, we put more effort in terms of cataloging. But some sample, they just want to curate it. Then there we just do a basic curation. Metadata matters. If you curate your sample, just packaging it. But there's no way people know the context around the sample, then there's no use of doing it. So the digital and the physical aspect is very important. Openness versus confidentiality. As you know, our program involves a lot of mineral exploration projects. So some of the samples are commercially in confidence. So using the system, we also have an access control by collection. So we can actually determine who can access and see the information about the samples. Those private samples are not exposed beyond the system to downstream applications. Stakeholder buy-in. So basically what I want to say is, based on my experience, the technicians who prepare the sample actually drive the curation. So we didn't hire someone to do the curation. Basically, it's just the technicians involved in the sample preparation were part of the project, and that really helped with the adoption, development of the guideline and adoptions, and also the leadership. So we had a strong, for example, we have updated the project proposal with all the guidelines and the system, which means that anyone starting a new project now will follow those guidelines. Invest in people. This is something related to what Kirsten mentioned. Instead of hiring a new role, we actually just train our technicians to do the digital curation. So because for them, it's just one step upskilling. Integration over customizations. So, for example, we want to have that system to identify sample using persistent identifier. We want the system to use some vocabulary. Instead of developing everything within the system, we just interface that system into relevant services. That includes the field collection app. So the current focus, we have a field mark. This is the field collection app used by the geologists to collect samples in the field. So we are interfacing our system with that mobile application so that when they get back to the office, they can just automatically transfer the data from the app. So that really reduces the need for manual data entry. So this is the integration, which I think sort of summarize. I think, yeah, thank you.
Thanks, Anu. And our last presentation before we enter the discussion is by Kirsten Elger, who will talk about the infrastructure and publishers' perspective on the sample life cycle.
Yeah, thank you very much. Maybe I must explain why I'm standing here. I'm not a publisher, but I'm Chief Executive Editor for Earth System Science Data, which is Copernicus data journal. And as such, I'm working closely with, I'm also involved in COPDES. So I'm now having a brief presentation on how are samples visible in journals and whatever. Just to recap, for those of you who have been here yesterday, the sample is at the lower end of the food web of research output. Publishers come in place on the other side, and normally this is where common practices show that IGSNs, and I'm affiliated with the geosciences, so I'm an IGSN user and provider, so that's where we are currently having practices to use IGSNs to publish the IGSNs on sample description landing pages and also to include them in data and to cite them in data publications. So they enable to connect data, well, IGSNs enable to connect data with samples, but how visible are they in practice? If I look at, I just did a little exercise. I did not look through all the publishers, but I checked whether there's any mention on sample provision, because we all know when you write a paper, you must provide the data, and there are increasingly guidelines to even provide access to the data upon submission of a paper. And they would not, like if you submit to any AGU journal, they would not even start the review process without having this access to the data. So they're getting extremely strict, but how about samples? AGU has a mention on IGSN, they recommend to use it, and if they use it, oh, hang on, this, I can't read it, so I have to go on. So they encourage authors to register samples with IGSNs and use them throughout the manuscript tables. So they really encourage them to use it. I don't see anything about citing them in, because why am I saying citing? Citing means that data should be cited in the references, and then we can appreciate or we can use the classical citation metrics to find the data citations, and this would be an equivalent to samples. So AGU is good, they mention IGSN. Nature is interesting, they are mentioned in policies of, in the Nature portfolio, they require materials to be available. With the publication of papers, they have a large focus on biosamples. So they give very clear guidance where you put in your DNA sequences or cell lines, including the facility Kerstin showed in her presentation. They support the RRID identifier, which relates to sample classes and not individual objects. So this would not be the case for IGSN. And if you look at other samples like geological or archaeological samples, they mainly provide a more general statement on make sure you preserve it, you report on your collection and everything, but without the specific notification on IGSN. The third publisher I've looked at, because I'm with ESSD, is Copernicus Publication, and you know that Copernicus is managing all the EGU journals too. So they actually require a sample availability statement if samples are registered with IGSNs, including the references. That is a very clear statement. And the lower example is actually from the HES data policy, which is one of the EGO journals, but they somehow use the very same general policies. But what are the challenges? I have personally seen the citation of individual samples with their IGSNs in papers on very rare occasions, but normally the paper describes not the results of one sample, but of hundreds plus. So citing 100 plus samples with their citations. Now we use IGSN through data sites, so they have authors, a title, publication. It's a classical citation. I have just checked it. I think citing 100 samples would require 5 pages of citations, and probably all with the same authors, because it's produced by the same group. This is that's not practical. That's the problem. A possible solution could be the newly developed and currently in development this complex citation object, where the idea is to provide a container that includes the required information that publishers can put in their cross-ref metadata to make sure that it's at least in the digital version of the metadata, but not on the papers. But this is something we can look into. And the other challenge, I think, is that IGSNs, despite being widely used and despite having almost 13 million, is still difficult to obtain. We have some national implementations. I think Australia is doing a lot to make this happen within their institutions. We have dedicated core repositories that offer them, but there's only one sample registration agent, which is CESA from Kerstin, where actually everyone can go and look. I know if and I see Rorie that you have put the IGSN registration functionality in your RSpace, but it's still not fully implemented. It should be closely related to the labs the samples come in. So it's this whole ecosystem that still needs to be developed. So that's another challenge for making samples more visible in the papers. And I just want to add that, of course, many sample repositories and also data repositories, what Kerstin said, we are not on the upper end of being funding. So we have temporary contracts, we have too little staff, but increasing expectations. What could work, what is currently happening, and this is just an example that could be one step to the solution, that we somehow collaborate with data repositories where the data are published. So that's an example from GFZ Data Services. It's a GeoRock dataset. We are currently having a collaboration with GFZ, with Digas GeoRock. So when samples are reported, we make sure, and you see to the left, that they are all cited in the data site metadata. That's what you see below. And also we link to the papers. So I think this is at least minimum a starting point where we get the samples through the data more into the papers. And that's it. I'm ready. Thanks.
Thank you. Thanks to our speakers for the different perspectives on the sample life cycle. Do we have a roaming microphone to take the questions and the discussion? I'm not sure whether anybody's online. I checked the Q&A. There weren't any questions yet online, but still we want to make it easy for anybody joining us online to hear the discussion. So we heard some lessons learned, but I'm also interested in hearing lessons learned in other disciplines. This was a very geo-heavy presentation series, but what's the situation like in other disciplines? Does anybody have any experiences to share, any lessons yet, or thoughts where they are thinking they might head? Nina, just wait for the microphone.
Oh.
Yes.
Hi, Nina Welty from CSIRO. I just have an experience at the far end when trying to create a federated data platform to expose data held in multiple repositories and harmonize it. We're now realizing that this sample curation at the very beginning is critical to making that very cool end stage possible. And the asynchronous investment across organizations is really challenging, and also challenging for these efforts to come together to say, I'm lacking and I need support in curation or ISG and minting or just a system. So I don't have anything to add except like triple underline, highlight, stars all around the importance of thinking about from the very beginning and identifying who does what, when.
I just wanted to add to that how important this issue is that Anu mentioned is the connection to any apps that make the collection of metadata in the field more streamlined or just simply easier. It's not a trivial task because field work is not always easy to integrate with digital equipment. A clear field where we need some more investigation. And yeah.
I want to just add about thinking later. I think the organization should recognize sample as a strategic research assets. If you don't recognize it, then you won't have the support for capabilities, even to adopt whatever process or tools. I know there's a lot of being pushed data as a research asset, but for me, if you ask sample, it's an important research assets, and that's the source of your data, right? So the organization should step forward to actually recognize it in their policies and how to do science.
Ooh, sorry, this sounds so booming in this corner. What the presentations also showed us is that dealing with the sample life cycle is heavily interwoven with all the different processes happening in research and collections management. Contrast this to, say, an ORCID as an identifier for an author. It's a very different world where the ORCID moves with the researcher. They are highly incentivized, if they care, to keep things updated. There are services to help them do that. On this end, where it's dispersed across organizations, across subunits, it becomes very difficult to build systems that nicely integrate, and the fieldwork component, a lot of that then speaks to we need to reduce friction in all those processes, otherwise that won't happen.
Hi, my name is Georgette. I'm the Senior Manager of the University of Adelaide Biobank, and I'm here on two capacities, but I'll speak on that one first. I just wanted to pretty much reiterate what the speakers have said. In the University of Adelaide, we had the stick. We were able to get our researchers to catalogue all their samples across at least the ultra-cold freezers, and these included not only the human space but also the animal and the environmental samples. But we were able to provide them with the infrastructure, not only physical, because we have a centralised facility, but we've also provided them with the infrastructure with a lab information monitoring system called Open Specimen. The training has been ongoing for the last, good 8 years. So we need to ensure that we tell our institutions and report the benefits or the risk versus benefit of having such a stringent and robust protocol. We've introduced a university-wide policy that mandates that the researchers need to do this. And it was all brought around because we had multi-million-dollar failures. Unfortunately, institutions won't support you unless they see that there is a benefit. So we've worked really hard on that space, and we're in a really good spot at the moment at the university, and we're also working with the state to ensure that we follow similar guidelines. One of the things that we're really trying to work on at the moment is standardising terminology, because as you mentioned before, it's really tricky to get all researchers to put information through in the same way. We have designed a template to try to mitigate that. But you've just said something about data and samples. So my other hat, which I'm wearing today, I'm the president of the ABNA, which is the Australasian Biospecimen Network Association, and we have our conference next week. One of our sessions is completely dedicated at data with samples versus samples with data. It is something that's really crucial in our space at the moment, and we have a debate on the Thursday. So I'll be sharing it on LinkedIn. But that is one of the biggest issues, at least in the biospecimen science, that we're seeing at the moment.
How interdisciplinary?
Take the mic.
Yes.
Can you repeat the question?
No, I was just asking how interdisciplinary or multidisciplinary is that effort to bring the specimens of a university into one consistent system?
Yes, so that's a very good question. It was primarily brought on from the human sample side of things. Having said that, one of the things that sets us apart is that we have been able to use, our LIMS is called Open Specimen, and we've been able to change the user-defined fields to suit all range of disciplines. So to put it into perspective, our biobank holds 50% of human samples, 40% of animal or basic research, and 10% of environmental samples. So we worked closely with the researchers to ensure that we had the right fields, the right terminology for each of the disciplines, and we're able to support them in that way.
I think we absolutely want to hear more from you. I don't know which.
Sorry. Seriously. No, just to follow on, if you say you have collected all the information, are these data open? So can others use this? Because that is an issue when we talk to biobanks, and there's a lot of personal data that is relevant and that they're not supposed to share. So, yeah. I don't know who.
Sorry. Yeah, so one thing that we have done, and we were able to do that within the LIMS, is to ensure that all the sample information is kept securely by each of the individual groups. But what we do offer is I can't remember what it's actually called now, it's like a catalogue, a generic catalogue. So researchers, if you're looking for a particular plant sample or if you're looking for liver tissue, you can go onto the catalogue and say, okay, so I need this group has got liver tissue, then you get in touch with the group and you can then liaise with the MTAs and stuff directly with them. So it is visible to our researchers, but without the particular information.
I just wanted to add some comment because my name is Liz and I'm the manager of a biobank for autism CRC. And the information there and the sample, we have data and sample is the identify. So there is no problem. We don't even have that information. It was kept in the research organization that collected the biological sample from this. And we offer through our services and go through agreement. But definitely one of the things that we want to do now, or we are working on, is to make the metadata more accessible, because if you are not able to see the data around there, people can use it, and that is, I think, is quite important. So.
I don't know if it's me or you, Ralph. I wanted to kind of build on some of the conversation around culture change and making things normative. At the top, we have making things required, big stick, incentivizing, so driving those incentive frameworks to make sure that samples are recognized as part of the scholarly record. And at the bottom, we have making things possible and easy from an infrastructure technology point of view. And kind of a question in that area is that, and we've learned this from a lot of kind of data citation practices, data is not cited. Data availability statements, in my opinion, are part of the problem. Is data available on request? Well, that's extremely helpful. So I guess the point I'm getting at is around data sets. What we've started doing is we've been working with various groups to go and actually work through corpuses of papers, and this is a bit of a tension with publishers sometimes. But being able to identify data citation, sometimes it's picked up in the method section of the paper, and infer the contextual information about that. Is it a primary citation of a data set, something that is directly created for that paper, or is it a secondary reuse of a data set that was created in perhaps another field and used as part of this paper? And a question, the specific question is anyone aware of or doing something in that context? I know RID do this, where they actually go and pick up RIDs in papers. Is there something happening in the IGSN context like this? Because I think that will help us move the making it easy kind of part of it, which helps drive we can incentivize it a bit more. I don't know if anyone's aware of any efforts.
Also, just introduce yourself briefly.
Sorry, Matt from DataCite.
Yeah, okay, Kirsten Elger again. Matt, I think the possibility to cite samples is there since the inclusion of IGSN as related identifier type back in 2016. And the rest depends on, I think it depends on the community. In the earth sciences, many journals would not publish the paper. Well, there are still exceptions, but from the repository side, I'm running a repository normally, we say, make sure you cite your paper, your data, and your sample. In the paper, this means include the citation, and the body text, and this reference in the references list. This is slowly going through. I still find even HEU papers where they say data available upon request, or some people who just add the DOI to the acknowledgement. But my argument to the researchers is they will never be found, which is true. And I think even there's an initiative in the UK who wants to have structured data availability statements. I don't think this makes a difference. So it is hard work, and it's really a lot of education to all parties. But if there is a support of, especially of publishers and of so it's very easy. They can, for example, they can check whether DOI is registered by using links to see whether it's not. And I know AGU is doing it, Copernicus is doing it, but I still receive emails from persons saying, hang on, look, my paper has been published 6 months ago. Why isn't the data, the link doesn't work? I said, yeah, well, because you haven't told me that. I'm not connected to the researcher, so to the publisher. So we must really count on the journals to say, okay, I do a serious thing, and the rest is education. Yeah, but Uwe is desperately raising.
Can I make one brief comment just on just one of the challenges, even if the publisher is capturing it in the paper, it doesn't always end up in cross-ref metadata and we miss it. And this is a huge challenge. Yeah, and so, but yeah, there's a lot of intricacies with cross-ref and things that we need to address, and relying on that is not a perfect solution.
Yeah.
Rolf Kral, Helmholtz-Zentrum Berlin für Materialien und Energie. I raised my hand because you asked for, say, inputs from other communities, although I don't have any experiences to share because everything I report is, say, plans and work in progress and not yet in production. Still, so we operate a synchrotron light source, a big user facility. So we are just in the process of setting up a central sample database for our facility with the aim to register all the samples that passes through, and also with the option to register IGSNs for them. And I can also report that in the photon and neutron community at the moment there's lots of discussions on how to deal with sample PIDs. I want to add one single remark because you were talking a lot about sample preservation and sample collections. Most of the samples in our research are created in the lab, measured and disposed after that. So they will not long-term survive. Still, I believe it is very useful to register identifiers for them because we want to track all the history of the sample from the creation to the characterization to the measurement to whatever happens to them, because that is the only way to get a decent sample description.
Just wanted to add to that from the perspective, is we have a lot of questions coming in from people who say, well, I used up my sample, do I still need to register it? And I keep saying, yes, absolutely, just because of what you said. You want the description to be there.
Hi, Uwe Schindler, Pangea, and also related to the Bremen core repository. So actually, about the citation and so on. So currently there's also a little bit of a problem because many IGSN currently registered do not really have author information, so a full sample citation is not really possible. So the question is also with the number of identifiers. So theoretically for most samples it could be okay if you have always a similar author, that you only have one citation with multiple DOIs. If that can be formatted by the authors, this would really solve that problem with this long citation lists going through several pages. But complex citations could also help. But are there any plans to improve the metadata? Because in the original IGSN proposals, there was only very, very few metadata, but no authors, no title, nothing like that, which would make a citation.
Come on Thursday to our session.
Okay.
Hi, my name is Susanne den Boer from the University of Copenhagen. I have a totally different question, which I hope it's okay. It's about sample safeguarding or sample security. I actually have a session on Thursday about value and a broader case from my university I would like to discuss, but now I have a room full of people who do sample management. I hope you have some tips. At our university, we're ramping up our activities relating to information security. I think that's normal for all of Europe. We have the threat of hybrid warfare, I guess, everywhere, but we can see that in increased activities safeguarding our research assets. So one of the activities that our information security team are doing in collaboration with the building administrators is, first of all, mapping how many freezers we have or where all the freezers are, which is a hellish task because many of them are purchased by independent research funding and we have no central overview. The second effort is to ensure that critical samples are backed up in freezers with, or are in freezers with diesel generators attached to them, right? Which now poses the critical question, what are critical samples? So we need to go out and ask our researchers, what is your valuable research? So that we can safeguard them against power outages, right? So now I've heard that you all are in the process of making guidelines. How do you ask your researchers to identify high value samples? Do you have some sort of classification, evaluation set up?
I think, we've...
Well, it might be the same answer. I was going to say Joseph Gunn has a repository scorecard that you can... Yeah, he presented yesterday in the... which could be used, because we were talking about it from our organization, as a risk management tool to identify when you're in that situation, what do you say first? Because it prompts you these questions and scenarios and stress tests your infrastructure so that as an institution, you start a framework. And he's around looking for a job.
I've known of activities in repositories, specifically this was the US Geological Survey, who created a decision tree on which samples to store and which ones to abandon. Aspects like, is it documented? Is there a label on it? Basic things, and then going to, are there data published about a sample, which makes it really, another level of relevance. And I've been using that actually locally in my institution to start building an inventory of the collections, just very similar to what you're trying to do, because the institutions struggle often with the space they giving to store collections. And if they no longer valuable because bags have fallen apart and all the samples are one big mess, you can get rid of it. So I think that is a really important incentive to getting institutions think more about samples. And I was so delighted to hear these efforts of a whole university taking samples as an asset and really trying to manage it. I think this would be a fantastic topic for one of the next sessions of this group to bring these aspects of, how do we get institutions to pay more attention, and how do we help them make policy decisions about the storage and the long-term preservation of them.
If I can briefly comment, what made our university get into action was a crisis at a Swedish university who lost millions worth of samples over Christmas. and because they could put some money on it, that could be taken to our university leadership and say, look, they lose this amount of money. We don't want to have that happen with us. So.
Any questions? I think I like the idea of putting a money value tag on samples, but I don't think it's sufficient, because we've seen enough cases in the geosciences where I have experience, where we know how many billions it would cost to recollect all the Antarctic samples, all the Arctic samples, all the ocean samples, and yet we heard how the Polar Rock Repository is struggling to secure continuing funding. It's the agencies who are caught in this tension between their mandate to fund projects, but their reluctance to fund infrastructure. It's not an easy win. I think in the cold storage of biological samples is a bit more of a hot topic than some rocks in a box. So areas for improvement, I think we heard a lot of points, but how do we stream this into something that becomes actionable?
Hi, I'm Lesley. I wear a couple hats too, and so ignore my shirt for right now because I'm going to talk from my educator perspective. Everyone keeps kind of mentioning education, but they haven't gone down to the next level below, which is the actual training of the next generation of geoscientists. And I know that most regions or nations have some sort of collective of university educators in the geosciences. So in Australia we have Algan, there's NACIT in the US, I'm not sure what the European equivalents might be, but it seems like going to them and making this this issue to the educators and saying, how do we work this into the curriculum, that this becomes part and parcel of field work of everything that students are learning from day one when they come in. And even, I work with high school students at the National Olympiads and seeing how it just starts becoming the mantra from the very get-go that obviously it doesn't fix the problem that we face today, but it changes the tide over time. Sorry, that wasn't really a question, that was just more of a comment.
Yeah, that's... I think there's a hand at the back. Very good comment, because this education piece helps to set the norms and expectations.
Many years ago, when I was doing my PhD, I used to have my lab notebook. However, that has changed now, and I think it would be similar that you can include due to intellectual property and that kind of thing, at least in molecular biology, people were asked if they have to have an official notebook. And now I think it's digital in many universities. So I think it could follow the same kind of process that you can do with the samples and be that official kind of form that people have to register all the information that can then be categorized and be in a generic form, so we can have comparable information and useful to be used.
And next here, but we tried to introduce lab notebooks in CSIRO, and we had very mixed success with that. And it depended.
It's different at universities, I think.
It is certainly different, but I would have expected a higher success rate at CSIRO, where you can mandate things top down, and it didn't happen. It had a lot to do with the requirements in the specific fields. So again, it points to the norms, where in molecular biology there are protocols that require better documentation than in other areas. So I think that we, again, come across these disciplinary differences, but also opportunities to learn from other disciplines how practices influence this culture change.
Just back briefly on the education piece, shameless plug. In January, we have a $600,000 National Science Foundation grant that's going to be convening a two-year two-year researcher program. We're going to be putting resources to support over 35 researchers. It's going to be open internationally, and we'll be taking them through a two-year course on professionalizing sample stewardship. workflows. We're going to get them out into their own collections, working with their collections, hearing feedback on what their challenges are, and then encouraging them to go present about their work in their own spaces. It's going to be interdisciplinary and open internationally. So I'll stick around after this session, and I'm happy to make sure that anybody who has researchers in mind gets that information when we release the applications.
I was thinking around what you said with the molecular biology. Sample management is really critical to IP strategy. So if you're thinking around the impact of research, and some organizations have this real big focus on commercialization or making use, and in order to have those contracts where IP returns a revenue back to research, in order to characterize what your IP is, i.e. your sample assets and what the data produced from those to build the models that get used in policy or apps or cool things. It can be quite, if you do this well, you have a very easy way to ring bind what an institution's IP is to generate future research revenue. So looking at it as a different incentive other than publication citations, but like it all comes back to the money, might be another layer to think around why the whole ecosystem has to kind of function towards it, because same way, not that I have a thought.
Hi, yes, Bryant Ware at Curtin University. I want to come back a little bit, or maybe it's still the same, but talking about field notebooks and or handwritten things. And with the CSIRO example of sort of even when you're coming top down, it's hard to get people to change their ways, particularly when you're dealing in the field, or just strictly using electronics in the field sometimes can be difficult. But I wonder, or want to, I guess, kind of quiz the room about using AI and instances like this to translate handwritten text into Word documents or something. I've been doing that a little bit with our Argonne lab at Curtin University because we have years and years of picking notebooks. The laboratory manager is hard to change her ways and that, but we have these very detailed picking notebooks, irradiation notebooks, and everything like that. And I've been using, trialing, kind of trying to use AI to look at those, and then you can say, just put it in a Word text. You could also tell it to put this in a nice table. I want to know what's the sample name, how much material was picked, and these sorts of things. Is that something that anybody's looked into or thought about?
No, I haven't used it personally, but I recently had a meeting with one of the group at the Berlin States Library, who is actually further developing these methods, and they look into handwritten information and old things from the 16th century and younger and older, to papyrus and everything. And what he showed me, what is possible, is just amazing, because you can identify. Well, there are open source tools. We can continue that later, but they're open source tools where you identify the fields of a form. And if it's our use case is very challenging because we have marine cruise reports with at least 3 different handwritings on every sheet. So we would need to train each and every one. So this is the question where but if it's a lab manager who's actually taking it, you do the effort in training to recognize this handwriting and to get information on. But it's amazing what is possible, I think.
I think that's a great suggestion that we all should look into. To remark on what we did with the electronic field notebooks is that they were most successful when we had standardized workflows, and then we had tremendous efficiency gains. The challenge that I always found in looking at workflows in research is that they're often quite ad hoc, and for a good reason. That's not that it's not rigorous, it's sometimes the discovery process that drives the protocol rather than the other way around. And that's where we struggle at the moment, when we deal with pre-structured tools. And so maybe AI gives us a new opportunity to become more flexible in having the research process drive the documentation, not the documentation drive the research. Do we have any other questions? Are we all getting ready for lunch? So we still have 10 minutes in this session, but we don't need to use them all up if everybody's now getting a bit low on blood sugar. But maybe we should spend a few minutes on summarizing the discussion and where this might be heading, and if we want to take this into the Research Data Alliance ecosystem of interest and working groups, because we have several interest and working group co-chairs here. Certainly for our session on Thursday on the samples, we already heard some suggestions of what we might pay attention to. Any other summary statements? You just get the mic.
It will be good to get some ideas about, I think you can use machine learning to extract information from papers, for example, that to go back to those old records. So some information about that, it would be great. Yeah, or to, some brainstorm.
I mean, I see some really important topics emerging, and in that respect wanted to, Jens mentioned the session that we're having on Thursday, kind of encourage people to join that session and to participate in that RDA interest group on physical samples, where, there has been, I think one of our challenges there has been that the originators of that interest group all came out of the earth sciences, and we've been broadening, but not enough, I think. And I've been really enthusiastic in this session to see, more contributions from biology, material sciences, or the overarching and institutional perspective on, a wide range of sample types in an organization. So it would be really great to have that broad perspective in that physical sample interest group as well.
Anything else? Any last questions? Just also, it's a shameless plug for Thursday. One of the contributions will also be from archaeology. We have a very active community of practice in archaeology and cultural heritage, but the archaeologists don't come to IDW. So we will have to make that connection by proxy.
Yeah, I think that is a really important point, that, it's hard to reach the right audience, of sample curators and researchers. So if you have suggestions how to integrate more with these different stakeholders throughout the sample life cycle, that would also be a really important discussion to have.
Well, I think then I might close this session. Thank our speakers, thank the audience for this fantastic discussion, and give you an early start into the lunch queue.